Let's welcome our next author, whose paper ID is 411. The presenter is Jihoon. Jihoon is a PhD student in computer science at KAIST in South Korea. He is a member of the Computer Graphics and Visualization Research Lab, led by Professor Gina Park. His research interests are biomedical image processing and analysis, currently focusing on the topic of 3D medical image segmentation. Now he will present his paper titled Hybrid Fusion Transformer for Multi-Sequence MRI. Welcome.
Yeah, thank you for a nice introduction.
Yes, you can share your screen.
Hello, my name is Jihoon, and I'm here today to present our research. The title is Hybrid Fusion Transformer for Multi-Sequence MRI. MRI is widely used in the medical diagnosis and analysis for various human organs. Capturing valuable information is one of the most important tasks for imaging. This is the reason that different MRI sequences are required. Each MRI sequence has different intensity distribution to the same target organ. These differences can make virtual result when combining multiple MRI sequences. Therefore, the way of how integrating multimodal images is the key point of multi-sequence MRI processing. We can divide the integration method into the time of fusion. Usually, early fusion and middle fusion method are used. Early fusion is the combining the raw data modalities, and middle fusion is the integration method of the intermediate results after some processing. Recently, deep learning-based methods have been successful in various computer vision tasks for natural images. Medical image domain also adopt the method of deep learning. Following the natural image processing method of early fusion, it means that RGB image is treated as 3-channel input. Multi-sequence MRI also use early fusion method like RGB images. But in the case of RGB-D image, consists of multiple modalities that have the same spatial information, color image and depth image. Considering the shareable and specific features between color and depth image, particular encoding is used for each modality in many tasks. We think that MRI sequences share the same spatial information but show significantly different characteristics for each sequence. Therefore, the early fusion method is not sufficient to fully utilize the multimodalities of multi-sequence MRI. To tackle this limitation, we suggest the hybrid fusion method that provides the advantages of both early and middle fusion method. And by constructing the novel hybrid network utilizing the locality of CNN and the global consistency of transformer, we propose the hybrid fusion transformer for multi-sequence MRI. The overview of the hybrid fusion transformer for 3D medical image segmentation is here. It is the case of for MRI sequences, and the CNN encoders could be changed flexibly depending on the number of MRI sequences. Our network could be divided into 3 parts. The first part is the multiple CNN encoders for hybrid fusion. The encoders consist of 2 types: the encoder with early fused input and the encoder for each modality. Multiple encoders generate the rich local const feature representations through the consecutive CNN layers. The next step is to utilize the global context of spatial information, considering the modality-specific information extracted from CNN encoders. All of the features from CNN encoders are embedded into 1D sequences, and then perform self-attention between feature embedding in the transformer layer. After the transformer encoder, we reshape 1D sequences to the 4D feature resolution and integrate them. As a result, we get the feature map that has the benefits of hybrid fusion. The remaining step is predict the segmentation map from the informative feature maps. Through the CNN decoder, the features are progressively upsampled and reach the original resolution of input images. During the upsampling process, we aggregate multiple features from the CNN encoders by skip connection for spatial information. We trained our network using both dice loss and crossfire entropy loss together. We used two publicly available 3D medical segmentation dataset consisting of multimodal MR images to validate the effectiveness of our method. BROAD-TS is the brain tumor segmentation dataset that has 4 MR scans of T1-weighted, T2-weighted, post-construct T1-weighted, and T2 flare. And MR-Brain-S is the whole brain segmentation dataset that has 3 MR sequences of T1-weighted, T1 inversion recovery sequence, and T2 flare. Comparing our hybrid fusion transformer with the 5 previous state-of-the-art models, including unit-based models and transformer-based model trans-BTS. In the quantitative result of 5-fold cross-validation in the BROAD-TS dataset, our method outperforms the other methods in all categories, except for the Hausdorff distance of the tumor core category. And the HF trans star, which is the variant model that consists of modality exception encoders instead of each modality encoder, also shows better result than the previous state-of-the-art. And we conducted further analysis through the qualitative comparison of the hard cases that all of the networks make coarse prediction result. We focus on the whole tumor region colored in green, which is an indicator of the ability to predict overall tumor shape. As you can see, NN-UNET tends to oversegment the tumor regions, and trans-BTS have reduced the oversegmentation, but it still remains, and even happens to undersegmentation in the first flow. In contrast to them, our method shows final result that capture the overall shape well. We think that hybrid fusion provide the ability to capture both powerful spatial context and modality context. The effectiveness of our method also can be found in the experimental result on the MR-Brain-S dataset. Particularly, brainstem segmentation result shows the benefits of our method. The grain regions in the dotted rectangles are the brainstems. More closely look at the regions, we can see the differences clearly. Most of the unit-based network have problems predicting only partial regions of the brainstem, not understanding the global context of brain structure. Trans-BTS makes better results than the CNN networks, but shows misprediction of undersegment or oversegment. Our hybrid fusion network shows perfect prediction with detailed boundaries in brainstem segmentation. It is evidence that hybrid fusion can understand the global context well. The proposed hybrid fusion transformer can vary depending on the encoder composition. We evaluate the various fusion methods from the traditional method of the early and middle fusion to the hybrid fusion, considering the human heuristics of the annotational protocol, which is that the appearance of brain tumor is typically depicted as a hyperintense signal in post-contrast T1-weighted sequence and T2 flare sequence. As you can see, the first law, the middle fusion approach, shows the worst result failing to get the benefit of each modality. When comparing the various hybrid fusion methods considering heuristics with the standard method of early fusion approach, they do not produce remarkable improvement. But the simple encoded composition of our proposed method, HF trans, shows better results without human heuristics. In addition, the variant of our method, HF transfer, also shows improvement in both metrics, especially with a remarkable Hausdorff distance result. To sum up, the proposed hybrid fusion method inherits the advantages of the early fusion approach with the powerful locality of 3D CNN and the middle fusion approach with the global consistency of transform. Experiments on different volumetric segmentation dataset, BRATIAS and MRBNS, validate the effectiveness of our method. The proposed method could serve as the basis for transformer-based segmentation network for multimodal medical images, with the fact that simple encoder compositions are sufficient for hybrid fusion. Yeah, thank you to all of the listeners. If you have any questions, feel free to ask me and contact us through our emails. Thank you.
Okay, thank you. Do you have questions? No, okay. Now we will have a break, and before that we will take a photo. So please open your camera.